Um, Hi, Hannah. Starting with Lab 21, isolation and identification of Staphylococci. It's known that Staphylococcus are gram-positive spherical bacteria which have the ability to form irregular clusters. So that's one identification for gram staining, which we usually perform. And there are different strains of Staphylococcus which is known to us, of which one is Staphylococcus aureus. It's also known as the MRSA strain, as it is the most prevalent type of the methicillin-resistant strain, which is formed due to the unhygienic practices in the healthcare systems or the medical field, right? Then, other than that, we have other common species called S. aureus, S. epidermis, and S. saprophyticus. So this lab is actually for identification to find out whether there is a presence of Staphylococcus if it's from two areas. One is from the nose as one is from the fomite. Okay, as a first step, what we actually do is we perform a sterile cotton swab. Two sterile cotton swabs are prepared, and we actually prepare a broth and try to inoculate into it. One will be labeled as nose, and the other will be labeled as fomite, of from the source from which it has been obtained. And we'll do what is done is actually as part of the second experiment, we do it is like streaking on the MSA plate, and then we look for the colonies being formed. So what actually happens is that when it is cultured on the MSA plate, two of the possibilities are there. One is that either it will be either a mannitol fermenting strain, or it can be that it does not ferment mannitol. So hence, if it's yellow, it's a mannitol fermenting strain of Staphylococcus, and if it's red or pinkish, then it's salt tolerant, but will not be fermenting mannitol. Okay, and if there is no growth, means no Staphylococcus has been isolated from the site or from the fomite area which we have been using for this experiment. So if you are looking out for the colonies, yellow colonies indicates it's positive, and here there is no fermentation of mannitol, hence it will be reddish in color. When there is no growth in MSA plate, it will be like with no colonies, obviously. All right, so moving on to the second experiment, we have inoculation on the blood agar plate. And we'll check for whether there is hemolysis of blood being added to it, because we actually have red blood cells added to the blood agar plate. If there is the presence of the enzyme which can actually hemolyze, then there will be clear zones being formed. If it's non-hemolytic, means the plate will remain red, means the red blood cells are remaining the same. So this is how it will appear. If it's hemolytic, there will be spaces where the lysis of the red blood cells have occurred. If it's non-hemolytic, it's going to be the same, as there will be, the plates will be red in color. If you can notice, even though the colonies are being formed, the red blood cells are not getting lysed. So that helps us to differentiate between hemolytic and the non-hemolytic strains. So you can say that hemolytic will be S. aureus and non-hemolytic will be S. epidermis. So moving on to the third experiment, it will be a staph latex test. What we actually do is we look for the presence of the enzyme coagulase. So red spots will indicate coagulation, and absence of coagulase will show uniform liquid. Means no coagulation will be present. So when it comes to staph latex test, this is how it will be present. If there is coagulase presence, then that means it's been coagulated. The lysis has taken place. In the absence of coagulase, it will be remaining the same. Then we have the obvious method, which is the gram staining, where we actually perform to see which strain is being used up. So this is how the Staphylococcus will appear. It will be cocci, as it's known. It's gram positive, hence purple, and the cluster formation is there. So purple, positive, and cocci, hence it's round. Okay, this is just a summarization of the lab 21, where actually we perform 4 tests. One is the MSA plate, then the blood agar plate, and the latex text, and the gram staining. Gram staining, you must be knowing about it. What you actually look for, if it's purple cells, it's gram positive. So since this is staphylococcus, it will obviously going to be purple cells, and hence indicating it's gram positive. If it's clusters, it's obviously staphylococcus, and if it's round, it's cocci. So giving these 3 tests as positive test. For the latex test, we are actually looking for the presence of the coagulase. If solid red dots or the coagulated form is present, means coagulase enzyme is present. If no dots are formed, then indicates the enzyme coagulase is absent. For the hemolysis test, we have actually red agar around isolated colonies if it's non-hemolytic. Clear agar colonies will be formed if it's hemolytic, means the bacteria burst the red blood cells in the blood agar. So if there is digestion of the red blood cells, we will get clear colonies, and if there is no change in the red color of the media, means it's non-hemolytic. Then for the MSI plate, as I said, if it's yellow agar, means it's a mannitol fermenter with salt tolerant. We're looking actually for 2 properties. And if it's not yellow, if it's red or pink, means it's just tolerant, salt tolerant, but not a mannitol fermenter. Okay, hope I made it clear. In the first test, we are only looking for staphylococcus. Just keep in mind these figures. If it's yellow colonies, what if it's pink or red colonies? What if no growth is found on MSA plate? Similarly for hemolytic and non-hemolytic. See, I think you can make out whether, since it's hemolytic, means there is digestion of the red colors and it's being absent in the non-hemolytic plates. Even though the colonies are formed, that's totally different from the basic blood agar plate, which we actually use for inoculation. Then coagulase test is one of the most simplest tests where we can look out for the presence of coagulase. If the coagulase is there, that means the digestion has taken place and clots are formed. If not, it will be negative. Then the gram staining, it's obvious since it's positive. Staphylococcus is gram positive, hence purple cells. It's round, hence cocci, and it's also seen as clusters. That's one of the basic properties of staphylococci when we do gram staining. So just keep this in mind. This will be the more helpful. Then go through the figures. That will help you to keep a complete complete picture of it. So that's what which is being given here also, even in your lab. And this is how you will be doing the descriptive chart. You'll check for the descriptive chart you have. You'll note down the observed results. Then you'll write down the inferred bacterial characteristics, how it's formed. If it's hemolytic, the coagulase is present. If the mannitol is fermented, that means it's SRS. If it's non-hemolytic, but a ferment mannitol, means, and also coagulase is absent, means it's S. saprophyticus. Similarly, if it's non-hemolytic, coagulase absent, but mannitol is not fermented, means it will be Staphylococcus epidermis. Please do keep this also in mind so that it becomes easy for you. Like you don't have to even think about it. You can just write it as such. So this is what we actually use for identifying whether it's from the nose or it's from the formite. You're actually checking out whether it's hemolytic, whether coagulase is present, whether mannitol is fermented, or you'll check for whether it's non-hemolytic, coagulase absence or presence, whether it's mannitol is getting fermented or not getting fermented. Okay, so I forgot to keep this tabular column in the lab. I'm adding that too, so that will be easy for you to brush up. Okay, so this is about the lab 21, where we have the isolation of staphylococci. Okay, just keep in mind, if it's hemolytic, then it is obviously SRS. If it's non-hemolytic, then there are 2 chances. It can be either mannitol fermenter or mannitol not being getting fermented. If it's fermenting, that means it is S. saprophyticus. If it's not fermenting, it is S. epidermis. These are the three strains for which we are actually doing this experiment. So I hope this much is clear regarding lab 21. Then for lab 22, it's actually the isolation and identification of streptococci. Okay, so streptococci is actually again gram-positive spherical bacteria that are arranged in chains. The arrangement of chains is 1 gram stain property of streptococci, which we can actually confirm directly. Okay, so streptococci, we have 7 major serologic groups: Group A, Group B, Group C, Group D, and even in Group D we have 2 subtypes. Then the viridians and pneumococci. If it's Group A, it will be beta hemolytic. You should be knowing this tabular column by heart, and it's S pyogenes. If it's Group B, these are the extremely virulent ones. These are the virulent among susceptible individuals. And when it's Group C, we have S. equi. Group D, which is Enterococcus fecalis, which is one of the major cause for urinary tract infections and other opportunistic infections. Then Group D, which comes under alpha hemolytic, again can cause urinary tract infections. And if it's viridians, it's again alpha hemolytic, but but it has a low pathogenicity, which is S. mutans. And the pneumococci, again we have the alpha hemolytic, it may cause pneumonia and meningitis with pre-existing conditions. So that will be identified as pneumonia. So you'll be actually looking out for which group the particular streptococci isolated will be belonging to. It can be either A, B, C, or the 2 subtypes under group D, or it can also be viridians, or it can also be the pneumococci one. But there are less chances of getting the pneumococci one because there are chances that you will be getting infected with this. So it can be basically from the rest 4 of them. No, the rest 5 of them, 5 or 6. Okay, so in this experiment, what we do is first is actually you try to obtain a swab culture from the back of the throat, usually to identify the streptococcal throat infection, and you try to perform a specific test to find out which particular strain is being actually located. So for the first step, what we do is actually you use a blood agar plate, and then we'll actually check whether it's beta, alpha, or whether it's non-hemolytic. So that will be the first step to identify whether it's beta, alpha, or whether it's non-hemolytic. Provided when you're given a streptococcal strain, that's the first step that you actually do. That means we'll check out whether it's alpha, beta, or whether it's non-hemolytic. So if it's beta hemolytic, if it's beta hemolytic, obviously the isolated colony will be the clear ones. Means they are able to burst the red blood cells in the blood agar plate. Similarly, if it's alpha hemolytic, means the isolated colony will be greenish in color. Right? If it's clear, means the digestion has taken place. If it's greenish, means it's partially bursting out the red blood cells. If it's the same, if it's remained the same, remain red itself, indicates that the isolated bacteria is non-hemolytic. For example, checking onto this. So beta hemolytic is always S pyogenes. You can see clear colonies in the areas where you have streaked. Along the streak lines, you can see that it's sliced off. when it comes to alpha hemolytic, you can actually see that it's slightly greenish in color, and there is partial digestion. If it's non-hemolytic, means it will be remaining the same. The red color of the blood agar plate will remaining this same or intact, as there is no digestion of the red blood cells being occurring in this experiment. Okay, so provided when you're given, if it's the first strain, provided if it's streptococci, if you get any instance of suspecting it to be streptococci, the first step will be inoculating on blood agar plate and checking whether it's for beta, alpha, or non-hemolytic. Then the second step will be on about 6.5% sodium chloride. And in that, what you actually do is that you're actually checking for salt tolerance, and that is usually done. These are the confirmatory steps for alpha hemolytic streptococci, right? So this is the first step. A few tests which you are actually performing for alpha to check whether it's, and if it's recognized as alpha hemolytic streptococci, then the rest of the test will help us to confirm whether it's alpha itself. Of which the first step will be 6.5% sodium chloride, where we're actually looking out for salt tolerance. So if it is positive for salt tolerance, means it will be clear. If it's clear, means it's not tolerant, and if it's cloudy, it means it's tolerant. Okay, that means the sodium chloride has been removed off. If it's clear, no, if it's cloudy, it will be the same. And what we actually do is we inoculate it first into a TSB media before inoculating it into the sodium chloride broth. Okay, so like I'm saying, it's a tryptic soy broth which you're actually using it for identification if it's alpha hemolytic. Sorry. Okay, so if it's alpha hemolytic, if you're performing the steps for alpha hemolytic, you check out the first step will be salt tolerance. In that, you inoculate it in a 6.5% sodium chloride broth. If it's cloudy, means it's tolerant. If it's clear, means it is not tolerant. Okay, and this is how the test tubes will be appearing. Okay, now I think I've got the wrong slide. Okay, then the second test will be bile esculin hydrolysis for streptococci. In that, what you actually do is you streak the surface of a BE slant with the help of a loop, which you have obtained from the TSP broth. What you actually do is that you take a culture from the TSP broth, which you did before, and you streak surface to a BE slant, or you do the slant culture for that. And here, if it turns black, then that means, yeah, it's the BE is being hydrolyzed, and that means the bile is being actually hydrolyzed. The selective agent, we are actually looking out for the bile inhibiting the gram-positive bacteria. Okay, if it's able to inhibit the gram-positive bacteria, means we can actually get a black BE slant. If the BE slant is not black, it means that the bile is not being hydrolyzed. You can check from the figure, the test and the control. The test is actually have turned a slant into black color, while the control is remaining the same. So control indicates a -1, and the test is actually positive for it. Means the bile has been hydrolyzed by the streptococci. All right. Then next test is the optogen susceptibility test. What we actually do is that we are checking for the sensitivity for that optogen disc. Okay, that's also called the Taxol. I think it's an antibiotic. So what we actually do is we keep it in the center, something similar to the antibiotic sensitivity test which we do in Kirby ball, and we are actually looking for the clear zone around the disc being placed. If it's sensitive, means there will be a clear zone. If there is absence of clear zone, means it's not sensitive. So that's what being is being given in the. There is growth and around the disc means it's resistant. If it's clear zone, then means it is susceptible to optogen. And similarly, we have the hippurate hydrolysis test, which is actually the next step for confirming whether it's beta hemolytic streptococci. What we actually do is we add sodium hippurate broth, and we'll check for the ability of the streptococcus strain to hydrolyze hippurate. So if it's able to hydrolyze, if it's cloudy, means if it is cloudy, that will really mean it's not being hydrolyzed, sorry. And if it's clear, it actually means hippurate is not hydrolyzed. Similar to something which you have obtained for 6.5% sodium chloride test. That means if it's cloudy, it's tolerant. If it's clear, it's non-tolerant. So this is about the optogen. This is how your results will be: a clear zone around the disc being placed shows shows susceptibility to optogen, while in the second half, it's the growth is seen around the disc, showing indicating that the strain is resistant and is not affected by the presence of the optogen disc. And in the hippurate hydrolysis, the purple color will be positive for hippurate hydrolysis. Here you can also check for the formation of whether being it's cloudy or whether it's clear. Though purple test color formation is not being given in your textbook, but you can check for cloudy or the clear test. And if it's cloudy, means it's hydrolyzed, and if it's clear, means it's not hydrolyzed. Then we have the next step, which is called the second test for beta hemolytic streptococci, that the bacitracin and XST sensitivity test. So here we are actually looking out for two zones, whether zones are formed around a bacitracin disc and also a clear zone around XST disc. Okay, if it's clear zone, means it's sensitive. If the growth is formed, it means it's not sensitive. Okay. Here again, and see, you can see the clear zone around the disc, and here the growth is not being affected. That means it's not sensitive, or it's resistant, in fact. And the one advantage of XXT and bacitracin sensitivity test is that A group and the B group are actually XST resistant, while the C, F, and G groups are XST susceptible. So that is also another way of identifying or one method to find out the particular strain which you have received from the streptococci, whether it belongs to group A or B, or it can also be belonging to C. So one parameter which you can use for checking is the bacitracin SST sensitivity test. You can specifically check for SXT test because the disc will be sensitive for the C, F, and G strains, while it will be resistant for A and B strains of streptococci. Similarly, it's not just XST, it's also maybe susceptible for the bacitracin. That means non-BS streptococci species strains will be susceptible to bacitracin also. Okay. And you can also know, you should be knowing about the full form of SXT. It's sulfomethazole trimethoform methoprim. You're checking out for the susceptibility for that. And this is one method in which you can differentiate group A and B from the other beta hemolytic strains of streptococci. All right. So The first three steps, hemolysis, 6.5% NaCl and BE hydrolysis, hemolysis is a common step which we do first to figure out whether it's alpha or beta. Then so salt tolerance, BE hydrolysis, and optician specifically to identify whether it's alpha hemolytic. Like if you get the first results positive for alpha hemolytic, you should be definitely carrying out these three tests. And if it's beta hemolytic, you should be carrying out hipurate, bacitracin, and SXT susceptibility. Besides that, you have one common test for all these, whether it's beta or alpha. That's the gram staining. And here again, it's in the chain because it's a strepto and it's gram positive, and hence it will be purple color. Okay, so that's about the overall about lab 22. Just keep in mind, if you're getting strains of streptococci, the first step will always be hemolysis to find out whether it's alpha or beta. If it's sodium then if it's alpha, you'll be choosing out for three steps: salt, BE, and optician. And if it's beta, you'll be checking out for hipurate, bacitracin, and SXT. Okay, so you will have to remember this. If it comes under alpha hemolytic, BE hydrolyzed, tolerant to NaCl, and non-tolerant to NaCl will be group D. If bile esculin is not hydrolyzed and susceptible to optocin, will be pneumococci, and resistant to optocin will be viridans. All right, I haven't added this. Let me add this too. And okay, so this is about identifying out the specific features of the alpha hemolytic. If it's alpha hemolytic and it's bile hydrolyzing and it's salt tolerant means it will be group D, while alpha hemolytic, bile not hydrolyzed and susceptible to optocin will be pneumococci, while alpha hemolytic, bile not hydrolyzed and optocin resistant will be the viridans group. Similarly, for beta hemolytic section, we have the three forms being identified based on the three different tests. So if it's beta hemolytic, like for the first step of beta hemolytic test, you get it positive. Then if it's SXT sensitive, it will be definitely group C. And if it's XST resistance, you'll be looking out for the susceptibility or resistance for bacitracin. If it's susceptible to bacitracin, it will be group A. If it's bacitracin resistant, it will be group B. So just study like this. If it's alpha, you have group D, pneumococci and viridans. If it's beta, you'll be checking out for XST sensitive and resistant. Similarly, looking out also for bacitracin sensitive and susceptibility, indicating C, A, and B. Okay, I hope it is not so confusing. You might have understood. And this is about the overall test. Initially, you can go for a gram stain to identify whether it's strepto or staphylo. And you do these three experiments if it's beta hemolytic, and these three if it's alpha hemolytic. And just after gram staining, you're supposed to be doing hemolysis just to be sure whether it's alpha hemolytic or beta hemolytic, and then follow up with the rest of the experiments. So if it's alpha, it will be salt tolerance, BE hydrolysis, and optocin susceptibility. If it's beta, you'll be checking out for hipurate hydrolysis, bacitracin, and SXT susceptibility. This is how you'll be looking out for. Whether it's clear or cloudy, it's the experiments are going to be, the results are going to be the same for salt tolerance as well as hipurate hydrolysis test. Those two will have similar outcomes, whether it will be cloudy or clear. And if it's cloudy, means it's not hydrolyzed, and if it's clear means it's hydrolyzed. Okay, then moving on to lab 23, where we have the isolation and identification of gram-negative intestinal pathogens. We have many species belonging to 5 genera, which we are actually looking out for in this lab. These are the common ones: E. coli, Proteus, Salmonella, Shigella, and Pseudomonas are few of the gram-negative intestinal bacilli. So you must be knowing each of them has to be gram-negative, but you'll also be looking out for gram staining method to identify which one belongs to which one. So for that, I'll start with the last. That means, like, when you do the gram staining experiment, if it's rod, it will be bacilli. If it's round, it will be cocci. And pink cells gram negative, purple gram positive, cluster staphylo, and chains streptococci. So these are, even though these are the common ones, you will be doing the gram staining for these strains also, just to identify whether it's E. coli, Proteus, Salmonella, Shigella, and Pseudomonas too. And as we know, like, these are some of the examples where which we have seen can cause food poisoning, enteric fever, urinary tract infections, bladder infections, dysentery, etc. So these are the strains which we are actually performing in lab 23 for the identification of gram-negative intestinal pathogens. So what you actually do is that in the first step, we streak plate it on Higton enteric agar, or HEA plate, and look out for the colonies, whether it's orange colony, green colony, or green colony with a dark center. These are the 3 outcomes when you do this experiment. See, if it's green colony alone, it will not be, there will not be production of H2S. If it's green colony with a dark center, indicates hydrogen sulfide production. If it's orange colony, if it's orange colony, you can just say that the lactose is actually being fermented. So on an HEA plate, you look out for three colored results. Like said, if it's orange, it indicates it's a lactose fermenting bacteria. And if it's green with dark colored center, it's a non-lactose fermenting that produces hydrogen sulfide. And if it is, it's green, it's just if the color of the colony alone is green, it means that the bacteria is not able to ferment lactose. Okay, I've just got it wrong. Okay, so this is how it's going to be. If it's green colony with dark center, it's non-lactose fermenting that produces hydrogen sulfide. If it's just the green colony alone, that means it's not able to ferment lactose. If it's orange, means it's a lactose fermenting bacteria. A bit strange, but you got wide variety of results for HEA agar plates. So I hope it makes it clear. If it's green colony alone, it's not able to ferment lactose. If it's green colony with dark center, means green colony specifically stands for not able to ferment lactose. So green colony with dark center will indicate non-lactose, but produces hydrogen sulfide. All right, and if it's orange, means it's a lactose fermenting bacteria, which we have actually formed. And how is this related to our final results is that we can actually look out if it's lactose fermented, means it is E. coli. Then the only possible organism is actually E. coli. If it's lactose not fermented, then we look out for the rest of the test. Okay, we'll go in the order so that it will be easy for you to understand. Okay, regarding HEA plate streaking, we have actually found out whether we're checking out for whether it's orange, green, or green with dark center. Then the second set of experiment is the Russell double sugar slant. Like if you have identified it as a non-lactose fermenting bacteria, then the next step will be, if it's lactose fermenting, you can confirm it's E. coli. Right, if it's lactose fermenting, you have actually confirmed it that it's E. coli. Of the 5 strains which we are discussing here, if it's lactose fermented, it's confirmed, it's E. coli. If it's non-lactose fermenting, we actually do the rest step, next step, that's a Russell double sugar slant. So in this, we check for the results. If it's a yellow slant with an yellow butt, means it's positive for the fermentation of lactose or even glucose, and it's causing the conversion of lactose to the formation of acid whenever it's yellow slant with a yellow butt. Okay, if it's red slant with a yellow butt, means it will be positive for the fermentation of glucose only, means fermentation of glucose alone. And if it's red slant with red butt, means it's neither fermentation of glucose or lactose. Okay, so that's how we are actually checking out for whether whether we are looking at, the experiment which we do for RDS only if it's non-lactose. If the isolated one is not is non-lactose fermented one, then we carry out this culture to identify whether it's glucose or lactose fermented, or whether it is glucose alone, or whether it's neither glucose nor lactose fermented. Right? So if it's glucose not fermented, then the obvious option is that it's Pseudomonas. Right? And glucose not fermented. Okay, and that's how we can confirm. If it's lactose fermented, we have the solution, it's E. coli. If it's glucose not fermented, then we have the option, it's Pseudomonas. Okay, so please do keep this chart also in mind and looking out for the next experiment. Okay, so just summarize this result. If it's yellow slant and yellow butt, means positive for both. That means glucose as well as lactose. And if it's only red slant and yellow butt, it's only for fermentation of glucose. And if it's red and red, means it's negative for both, neither glucose nor lactose fermenter. Okay, that's one way we can identify between the non-lactose fermenters. If the glucose is not fermented, you can write it out that it's Pseudomonas. If it's lactose being fermented, we have the result as E. coli. Okay, then the next step will be the urea broth culture, where we are actually evaluating the motility of the strain. Like for that, it's quite interesting. Like we actually do a stab culture and we check out whether there is growth beyond the stab. If the growth is only along the direction of the stab, means it's non-motile, means it's only along the direction of the stab. If it's motile, means they'll have a diffused edge, in the sense that they are able to grow widely across by using the nutrients from the media. And if it's motile, means, and also there will be clear turbidity being visualized, because the microbes are able to survive and replicate within the media successfully. So think like this: if it's non-motile, means the growth will be along the stab line. and for the rest there will be a diffused edge and also a formation of turbidity. So how is this related to the chart given to us? If it's non-motile, we have Shigella as the example, right? So we have three examples. Glucose fermented, it will be E. coli. Glucose not for lactose fermented, it will be confirmed as E. coli. Glucose not fermented, it will be Pseudomonas. If it's non-motile, it's going to be Shigella. Okay. Hope it's clear. Though you have to keep that in mind regarding them. Okay, then what you do is the next step, which is the urea hydrolysis. When do you do urea hydrolysis? Where is it? Okay, what happens in urea hydrolysis is that you're checking out whether the presence of the enzyme urease is present. If there is no color change, means or if it's yellow orange, means its enzyme is absent. If the enzyme is present, the urea hydrolysis has taken place and there will be a formation of bright pink color. And you can also keep in mind urease positive ones are Proteus and Helicobacter. So in this case, you can confirm it as a Proteus species. Let me see, where is the urease test? Okay. As I said, for motility, if the growth is within the narrow stab, then that means the bacteria is non-motile. So here we can confirm it's Shigella. And when you're evaluating urea hydrolysis, if the color of the urea broth turns bright red, pink, or reddish pink, that means the bacteria has hydrolyzed urea. If there is no color change, then the bacteria is not able to hydrolyze urea. Okay, that's again is given here, but you can check for, if it's motile, there are two chances, urea being hydrolyzed and urea not being hydrolyzed. So if it's urea hydrolyzed, you can confirm it's Proteus. If it's urea not hydrolyzed, you can confirm it's Salmonella. Right, so this tabular column is again very important in identifying which is a possible strain given. So you need to keep that in mind. If it's lactose fermented, done. The first step is that it's E. coli. It's confirmed to be E. coli. And if it's non-motile, you can confirm it as, if it's non-motile and it's glucose fermenting, you can confirm it's a Shigella. If it's glucose fermenting with motile properties, you can check for two other properties, urea hydrolysis and urea not being hydrolyzed. So if it's urea hydrolyzed, it will be Proteus. If it's urea not hydrolyzed, it will be Salmonella. And if it's not, as I said, if it's glucose not fermented, then it will be Pseudomonas. So it's a connecting way in which one after the other we do a sequence of steps to identify whether it's lactose fermenter or whether it is a non-lactose fermenter, whether it's motile or whether it's non-motile. So all these steps are interconnected. So only if it's glucose, only if it's non-lactose, or like if you get it as only the glucose as the only fermenter, that's the step when we do motility test as well as the urea hydrolysis test. Okay, that will confirm whether it's Proteus, Salmonella, or Shigella. If it's non-motile, it's going to be Shigella. If it's urea hydrolysis, if it's hydrolyzed, it will be Proteus. If it's non-hydrolyzed, it will be Salmonella. So the best ideal way will be to learn this taxonomic key as well as the possible observed results which you get for every single experiment. So with that way, you can figure out which strain is going to be present. And even though even if your instructor just send you the culture, you'll be able to look it out, and you'll be able to infer the causative agent or the disease pathogen present in it. If it's like I said, so everything is interconnected. So it will be ideal if you can remember these slides and this slide, the cost of agents, and the one how you infer by reaching into it, and the specific test. So I'll say for lab 23, these two, the last two slides will be important. For lab 21, the last three slides will be important. And for lab 21, you have the other set of tests for that. I think I have not added that. I think I've added. Lab 21. Okay, I've added. So these slides will be the important ones for lab 21. So if you can just keep these in mind, it will be easy for you to remember. And keep in mind regarding the specific outcomes, which will be easy for you to understand. I'll send you the possible questions and the possible practice set of questions which you can do. Let me just search for a while, and I'll put that on a Word document and I'll send it to you.